Okay, well, thank you very much for the introductions. Uh, so today I'll talk about the real-time IntraVital microscopy system from IVIM Technology as your in vivo live animal cellular imaging platform. Okay, so as some of you may already know, the IntraVital microscopy is a very unique technology that can provide a very detailed live video in cellular level in a live animal model. So in this example in the middle, what you are looking at is the lung of sepsis mouse model. The green shows the blood circulation, which is visualized by intravenously injected fluorescent dextran, PT-dextran. And the red color is actually intravascular neutrophil inside the blood vessel. They are also fluorescently labeled by antibody conjugate. They can specifically label the neutrophil. So in this particular example, we injected anti-Lysex antibody conjugate with the red fluorophore intravenously at 3 hours before this imaging. And those intravenously injected fluorophore antibody conjugate specifically label the neutrophil and washed out from the blood circulation. And then we do this IntraVital imaging of the lung. So in this example, what you can see is this movement of the neutrophil, very active movement of the neutrophil inside the lung of sepsis mouse model. And as you can see, at some point, this neutrophil make a cluster inside the blood vessel, and then it completely disturb the blood circulation in this area. So this is a very good example that IntraVital microscopy can provide a very variable live imaging in the development of disease. So IVIM Technology's IntraVital microscopy can enable this kind of very dynamic 3D imaging of various cellular level dynamics, such as cell trafficking, cell-to-cell interactions, and cell-to-microenvironment interaction inside the living preclinical animal model in vivo. And for the drug development, also this IntraVital microscopy can enable a direct imaging analysis and monitoring the drug delivery to the target tissue and cell, and also assess the drug efficacy and validate the mode of action in also various preclinical animal model. So I hope you can see this video. So on the left side, this is the IntraVital imaging example obtained from the lung. In the middle, what you're looking at is the transplanted bone marrow cell in the bone marrow of the mouse model. On the right side, what you're looking at is the granulocyte trafficking in the skin during the inflammatory response. So this kind of very dynamic cellular level movement can be directly imaged by using IVIM Technology's IntraVital microscopy system. And I hope you can see this video as well. So this is also example of the monitoring of the drug delivery. So on this left side, what you are looking at is the delivery of the nanoparticle to the liver in real time. So in this example, this red fluorescent nanoparticle was intravenously injected at the start of this video. And then you can see the arrival of this nanoparticle through the blood circulation and accumulation in the liver in real time. In the middle, what you're looking at is this red dot is actually drug carrier, or again, intravenously injected, which delivered through this blue color vessel to this green cancer cell in the cancer genomic model. In this left side, what you're looking at is the adaptively transferred T cell to target the cancer cell in triple negative human breast cancer xenograft model. So you can see this interaction of this red T cell and the green cancer cell in vivo after the injection of these T cells. So these are the good example of in vivo testing of the drug. So IVIM Technology's IntraVital Microscopy system have been applied for almost all the tissue and organs, as you can see in this slide. So the bone marrow, brain tumor, retina, skin, the hair follicle in the skin, and then tumor, actually this is colorectal tumors, pancreas, spleen, stomach, and GI tract adipose tissues, prostate, placenta in the pregnant mouse, and various lymph node, memory tissue, and then kidney transplant, and skeletal muscle, liver, heart, lung, pleura in the lung, and the trachea and thyroid. In addition to this, we can also see the blood vessel, the microcirculation, and also the lymphatic drainage as well. So almost all the organ and tissue can be imaged, can be visualized in vivo with these very high resolutions, as you can see in this slide. So IVIM Technology's product, their microscope line include confocal system for IntraVital Imaging and two-photon system, IVIM M model, and then dual mode confocal and two-photon model as a CM. And then we recently released in this year the two-photon microscope dedicated in a very compact two-photon microscope system in the market. And then we also provide research service as well, who interested in using the IntraVital imaging for their research. And then some unique features. The IVIM Technology's IntraVital microscopy model have unique features such as all-in-one single box packages, as you can see in here, for easy installation and operation and maintenance. And then all the hardware and software is co-optimized for IntraVital imaging. So it can also be used for standard conventional tissue observation and tissue slide observation as well. But additionally, it's dedicated and optimized for the IntraVital imaging of a live animal model. So it has unique features such as ultra-fast real-time imaging, up to 100 frames per second. And then it also has live tissue motion compensation functions. And then it integrates all the device and apparatus for various organ imaging, and then to keep the animal alive during the imaging. So basically, as you saw in the previous slides, there's almost no limitation in imaging various internal organs. So the imaging speed up to 100 frames per second. Standard version is actually 30 frames per second, but with 512 by 512 pixels per frame, the IVIM Technology's IntraVital Microscopy confocal and two-photon system can enable very high-speed imaging by using the rotating polygon meter. And this rotating polygon meter has additional advantages, like it can easily achieve the uniform excitation illumination over the entire imaging field of view. In comparison, the conventional resonant galvanometer scanner suffers the reduced presence in the middle, at where the scanning speed is much higher, and then it has excessive photo bleaching at the edge area where the scanning speed is slowed down. So, but in contrast, by using rotating polygon meter, we can achieve the uniform, very high speed imaging in the entire field of view, up to 100 frames per second. And then another unique feature of the IVIM Technology's IntraVital Microscopy system is automatic motion artifact compensation functions. So in real example, in real situations, when you look at the internal organ, we always see this kind of motion because of the breathing and the heart beating. So even though the mouse is the animal is anesthetized, deeply anesthetized, so there is no muscle, movement, but still, the heart is beating, and then it should breathe air, and then those physiological motion create tens of micrometer to hundreds micrometer, motion during the imaging. So this is actually the real-time imaging of the kidney, and this is very typical movement of the tissue when you do this IntraVital imaging. And the IVIM Technology's IntraVital Microscopy system has automatic, hash-free, high-precision motion compensation functions. So in this, as you can see in this example, from this real-time video, the IVIM Technology's IntraVital Microscopy system can generate this motion compensated imaging by averaging the multiple frames. And then without this motion compensation functions, and then with the slow imaging speed, you will always end up this kind of blurred images from this moving, fastly moving tissues due to motion artifact. And this is another example. So this is, another example of the tissue motion in real situations. So what you're looking at is the blood vessel and then CXCCR1GFP cells in the heart, actually. So without the motion compensation functions, you will always, end up this kind of blurred images. So you definitely need frame by frame motion compensation to achieve this high resolution imaging from this, moving tissues. So this is the real capture of our software to how it works. So from this kind of movement, what you have to do is just activate the motion compensation functions and just press the image captures. Then you can instantaneously get this motion compensated imaging from this real-time video. So in the middle, what you're looking at is the real-time averaged images. So and then this is what you will get without the motion compensation. But with this automatic motion compensation function, we can guarantee you can get this higher resolution motion compensated imaging from this kind of tissue movement. And then we also provide the body temperature monitoring and feedback heater control system as well. So we provided this electric probe to monitor the body temperature, and then this plate for the animal imaging has integrated heater inside. So we monitor the body temperature, and then this and using with the feedback control, we can keep the body temperature to be stable. So this is the heater temperature and body temperature, and then we also provide additional temperature sensor to monitor the tissue temperature during the local tissue temperature during the imaging as well. So in our system, we have the front door, and then if you open the front door, and then you can slide out this plate holder, and then you prepare the animal for the imaging, and then put it like in the middle, like this in the middle, and then you put it back, and then place the animal under the objective lens for the IntraVital imagings. So when you open this slide door, you will have this kind of layer and then sliding plate holder to place the animal in the middle. Okay, so from now on, I'd like to explain a little bit about the technology. So first of all, I'd like to explain a little bit about the confocal microscopy model, the IVIM Technology's IVIM-C and CM model. So I guess many of you already know what is confocal microscope. Confocal microscope is optical sectioning microscopy to get the sectioned image from the thick tissue. So by using the confocal pinhole in front of the detector, we can effectively detect block the presence originated from the out of focal plane, like this. And then using this pinhole, the fluorescence originated from the focal plane will pass through this pinhole and then detected by the fluorescence detector. So by doing that, we can selectively detect the fluorescence from the focal plane. And by doing that, we can get this higher resolution sectioned imaging from the sick tissue inside the live animal. So this is the real data we obtained from the immunoadipose tissue in vivo in anesthetized mouse. So in this particular example, we used the adiponectin Cree crossed with the M2MG mouse. So basically all the adiponectin expressing adipocyte has a green membrane. So this green is actually adipocyte. And then all the other cells express the TD tomato, membrane tomato. So basically you can see the vessel and some other stroma cell in red color. And this is 4 micrometer G-stack images obtained from this immunoadipose tissue, again, in the live animal model. And from these G-stack images, well, we can easily get this subcellular resolution, 3D rendered data to, analyze the tissue in subcellular level in vivo. And the Topo Microscopy is another technique for optical section imaging in the live tissue in vivo mouse model. So in Topo Microscopy, the fluorescence signal is intrinsically generated only from the focus. As you can see here, for 2-photon excitation from this right side, you can see fluorescence signal is only generated from the focal plane because of this 2-photon excitation. So compared to the single photon excitation used in confocal microscope, so the entire volume is fluorescent. But in 2-photon excitation, well, you can easily achieve the optical sectioning. And then by using the longer wavelengths, near-infrared pulse lasers for 2-photon excitation, we can achieve deeper tissue penetrations. In addition, this 2-photon microscopy is capable of label-free nonlinear multi-harmonic generation imaging, such as second harmonic generation and third harmonic generation imaging. So, and then this second harmonic generation is, quite interesting, imaging modality, which can be easily achieved by 2-photon excitation. In biological tissue, the collagen has a strong second harmonic generation signal. So, in other words, the collagen and some microtubule and regular protein fibers can be detected without any additional labeling by detecting this second harmonic generation signal, which can be easily performed by using our 2-photon microscopy system. So this is the good example of the pivoted collagen in the liver, again obtained in vivo in the liver of the mouse model for non-alcoholic fatty liver fibrosis model. So you can, in green, you can see the pivoted collagen detected by second harmonic generation, and additionally, you can see the fluorescence from the lipid droplet by using fluorophore. So this, actually, this is the maximum intensity projection images from the G-stack data. So this is the 2 micrometer interval, G-stack images obtained from the liver of live animal model of fibrosis and steatosis. And also, this second harmonic generation signal is also strongly detected from the sarcomere of the myocyte. So this is actually the mosaic image obtained from the thigh muscle of the anesthetized mouse. So in this particular example, actually we use Cy1 YFP mouse. So this green is actually yellow fluorescence protein, which expressed in the nerve axons and this neuromuscular junction inside the muscle. So in red color, actually this is a sarcomere in the myocyte. So you can see this periodic structure inside the myocyte. And then so which is detected in label-free manner by detecting the secondary generation. So this is micrometer G-stacks. And then this is the maximum intensity projection images. And in these images, actually we label the blood vessel as well, again, in vivo by using anti-CD31 antibody conjugated with the fluorophore. So in this example, we injected this CD31 antibody conjugated to the fluorophore intravenously at 2 hours, with 1 or 2 hours before the imaging. And then it systemically label all the blood vessels inside this transgenic Cy1YP mouse. And then with 3 color, using secondary generation and YFP and CD31 process antibody, we can detect the microvessel inside the muscle. And then this yellow process protein expressed neuromuscular junction, and then myocyte in the inside using the secondary generation signal. So IVIM MS2 is actually the most compact and affordable IntraVital two-photon microscopy system in the market, actually. So it fully integrates with the very highly compact femtosecond pulse laser module for two-photon imaging. So in here, we has this femtosecond laser module inside it for two-photon imagings. And then this whole box, this main box size is only 500 by 620 by 470 millimeter in width, depth, and height. And then it also has this sliding blind curtain to completely cover this sample stage. So we don't have a requirement for the dark room for this microscopic operations. So instead of using conventional titanium sapphire femtosecond laser system, which is very expensive, by using this compact pulse laser, femtosecond pulse laser module, we can cut down the total system cost by half compared to the conventional two-photon microscope system. And again, by using this IVIM MS2 model, we can easily obtain this higher resolution, subcellular resolution IntraVital imaging from various tissue like skin, lung, skeletal muscle, and also body tissue, and lymph node and other tissues as well. And then we also provide the image processing software as well, the IVM Studio, for 2D, 3D, and 4D imaging data visualization and analysis. So this is the 3D rendered data from the small intestine actually. So you can see the XY and YG images together with the 3D rendered images. And this is a 4D data set, basically time-lapse 3D G-stack imaging data. So you can see the movement of the cell in the tissue, in the live tissue of the anesthetized mouse. So again, you can see this oblique, and then you can also see this sectioned images in 3D from this G-stack time-lapse 4D data set. And of course, you can see this kind of 4D data set to observe the movement of the cell in the live tissue. So this kind of very high resolution IntraVital imaging can be obtained by using the IVIM Technology's IntraVital microscopy system, the two-photon and confocal microscopy system. And then I'd like to show you the real, some example of this various tissue imagings. So this is the preparation for the liver imaging actually. So well, you need to make some incision on the skin, abdominal skin and peritoneum, and expose the liver, and then put the glass cover to seal the exposed area. And this is the real-time IntraVital imaging data obtained from this setting. So in this particular example, we use the type 2 GFP mouse. So all the liver endocellular cell express GFP, so we don't need to process labeling. And then you can see this intravenously injected nanoparticle inside the liver sinusoid, as you can see in this video. So you can see this flowing nanoparticle in the liver sinusoid, and this nanoparticle attached to the liver sinusoid endocellular cell in real time. So actually, this is just a 20-second real-time video obtained from the liver right after the injection of this nanoparticle. And then on the right side, actually this is a 4-minute video to show the uptake of the hepatocyte of the intravenously injected fluorophore. So in the beginning, you can see that this arrival of this fluorescence probe through the blood vessel to the liver only within several tenths of a second. It fully filled up the blood vessel, it was sinusoid, and then it gradually leak out from the liver sinusoid and then uptake by the hepatocyte here, like this. So this is the real-time uptake monitoring in the tissue. In this case, it's liver. And then also, after this, we can monitor the clearance as well. So as you can see here, up to 6 hours, we monitor the same site of the liver to see the clearance of this fluorophore from the liver. And then the another, beauty of the IntraVital microscopy is, we can keep the animal alive during the imaging, and then we can also save the animal after the imaging. So it means we can repeatedly image the single mouse model over a very long period of time, up to several days, from several days to several weeks to even several months. So this is the one example of this longitudinal imaging to see the disease development in the live animal model. So in this particular example, we created a non-alcoholic fatty liver by giving the MCD diet to the live animal model. And then with this diet, this mouse developed the fatty liver very rapidly. So only 2 days after of this MCD diet, you can see the development of this lipid droplet inside the liver. And then, with the continuation of this MCD diet, we can see the increase of this lipid droplet size in the liver of the live animal model. So in this example, the lipid droplet is fluorescently labeled by using SF44, which is some lipid droplet specific fluorophore, which was intravenously injected right before the imaging, actually. And again, the liver sinusoid was labeled by using anti-CD31 antibody and fluorophore conjugate. So this is just the same data from day 2, 7, 14, and 21. And then, in addition, as I explained to you before, by using the second generation imaging mode, we can detect the collagen fiber as well in the liver fibrosis model. So in this example, we make the high-fat diet model. Using the high-fat diet, the prolonged high-fat diet, the mouse spontaneously developed the liver fibrosis. And then you can see this green, actually fibrotic collagen in the perisinusoidal area in the liver. And at the same time, you can see that this formation of this large lipid droplet inside the hepatocyte. And this is the 3D reconstruction, of course, after taking the GSTAC data. So in this example, you can see that this development of the fibrotic collagen in green, together with this green red hepatocyte, red lipid droplet in the live animal model in the liver. And then, we also can provide the imaging support, experimental support from our application specialist. So actually, this is the video we uploaded after we recorded it in our company, the IVIM Technologies. So actually, this is the surgical procedure for the dorsal skin fold imaging chamber, which is well-established imaging chamber device for the cancer drug imagings. So basically, we expose the subcutaneous tissue, and then we can inject the cancer cell, and then seal this subcutaneous area. And after this setting, we can monitor this cancer-derived model for over several weeks, more than 2 weeks, 2 to 3 weeks. And then in this setting, so by using our real-time imaging capability up to 30 frames per second to 100 frames per second, we can easily see this flow of the red blood cell in this genomic model actually. So in this particular example, to show you the better contrast, we use the pseudo color for the red blood cell in green. And actually this blue is actually the cancer cell expressing the GFP. So this is the real-time data to show the cancer cell, and then tumor vessel and the cellular cell, and then IVIS flow inside. So basically, this is the data to show the tumor vessel and then tumor vessel perfusion with the red blood cells. And then again, in this setting, we can keep the animal to be stable for a very long time. So this is actually the monitoring of the nanoparticle delivery to this triple negative breast cancer cells, MDMB231 GFP model. So in this setting, so this is 2 hour, 6 hour, and 24 hours imaging at the same site after the injection of this nanoparticle. So at 2 hours, you can see that some nanoparticle, and then at 6 hours, you can see much more increased delivery of this nanoparticle through the this blue tumor vessel. And then finally at 24 hours, you can see the diffusion of this nanoparticle from out of this tumor vessel, and then delivers to the cancer cell. Well, for example, here, and here, and here. So this IntraVital microscopy is a very good technology to monitor the delivery of the drug. In this case, it's nanoparticle, but it can be also used for other type of drug as well. And then this is the 3-day interval observation of this cancer drug model to see the cancer vessel remodeling. So this is actually the same vessel with 3 days interval by using this dorsal skin fold chambers. So you can see the infiltration of this GFP expressing cancer cell and the remodeling of the tumor vessel. As you can see the huge dilation of the tumor vessel together with the infiltration of this cancer cell over the course of the 6 days. And another very well-known technique for the long-term IntraVital imaging of the brain is this cranial imaging window. So, so this is again the video we uploaded on the YouTube. So we remove the small area of the skull and then put the transparent cover glass, and then put the UV adhesive to seal the area. And then after this setting, this mouse can be observed for more than several months. And this is the real data we obtained from this setting to show the astrocyte and pericyte and the blood vessel together. And then in this setting, actually, this is how we this is the just one example experiment we have done. So after we implant this cranial window, we normally give the mouse for more than 2 to 4 weeks for the recovery from the surgery, and then we induce the neuroinflammation by injecting the 3-MP chemical, and then we longitudinally observe the same animal for more than 30 days. And in the first 10 days, this is the example. This is the observation of the same site in the brain through this cranial imaging window. So you can see the infiltration of these RIGMGFP expressing cells in the brain. So you can see, as you can see, we can observe the same area with 3 days interval, and then we can identify the same area by just looking at the blood vessel. Okay, and then we can see the change of this cell distribution and the cell number in certain area of the brain. And then of course we can do this dynamic imaging to measure the cell movement in this area. And then this is actually 30 days observation of the same animal in the brain cortex. So you can see this specific blood vessel morphology here. So this is actually same site. You can see. So this side here and it's here. And then, okay. So what's interesting in this observation was in the beginning there was no blood leakage in the beginning. So it was monitored by advanced blue, intravenous injection of advanced blue. At day 7, there is no leakage, and at day 14, suddenly this area had some vascular leakage. And then this vascular leakage was just sustained more than up to 21 days. At later time point, we can see this infiltration of RIGMGFP expressing granulocyte in this area. So BBB dysfunction followed by the infiltration of the granulocyte can be monitored in the same mouse over the time course of 30 days. And this is another YouTube video to show the surgical preparation for the IntraVital lung imaging. So we do the intubation, and then we use a ventilator to keep the mouse to breathe during the imaging. And this is the stabilized imaging window for the lung observation. So we use the negative air pressure to stabilize the small area of the lung. And after this stabilization, we can obtain this high resolution image from the lung of this live mouse model. So, this was the example I already explained. So this was the real data we obtained from with the setting I showed in the previous slide. And then this is actually the real-time, IVIS flow images, and then with the IVIS tracking analysis, and then we can see this blood vessel in the lung, and then also we can monitor the IVIS perfusion in this particular area of the lung. And then actually by using this same device, we can actually observe the heart as well by using the negative pressures. So again, the certain part of the small part of the heart is stabilized by using this imaging window chamber. And then even after this negative pressure, we still have this huge motion. But as I explained to you, we have automatic motion compensation functions. So from this real-time video, we can get this high resolution, average the highest image data using motion compensation function. So this was just some of the example what you can do by using the IntraVital Microscopy system. And then we also provide the IntraVital imaging service for you as well. So this is the procedure basically. So we provide consulting for the imaging experimental project, and then do the preliminary test, and then for whether this imaging experiment is possible or not. So validate the imaging plan, and then optimize the imaging parameter. After that, we propose the optimized imaging plan with the quotations. And if you accept, then we do all the imaging procedures. So prepare an animal model, do the imaging, and the processing and analysis, and then provide the report. So these are the parameters we have to consider how to do the in vivo labeling, and then how to do the surgical procedure for the specific certain organ imaging. and then what's the imaging parameter we need to achieve the imaging goal, the experimental goal, and then what's the animal model we need, and then do how to do the real procedure for the IntraVital imaging and then analysis. And this is the one of the our recent imaging service result, which was published in this paper actually. So this is the one data we provided to our customers. So in this example, we basically monitor the delivery of the exosome. So in this example, only 10 minutes after, you can see this red color exosome, was attached to some specific cells in here. And at 30 minutes after the intervention injection of exosome, it was actually internalized into this particular target cells. So again, this is the very good example to how to utilize the IntraVital microscope to monitor the drug delivery. In this case, it was exosome. So yeah, so IVIM Technology is providing this all-in-one, highly optimized IntraVital confocal and two-photon microscopy system with the motion compensation functions and the animal model, control and maintenance device. And then we also provide IntraVital imaging service, R&D service for you, if you're interested. And then with more than 10 years of the experience in this IntraVital imaging experiment, we have a specialist team with advanced know-how to how to do this IntraVital imaging in various organ and tissue in live animal model. So we can provide the consulting support for IntraVital imaging experimental design, and we can also provide the technical support to validate what will be the best way to label the specific cell or specific tissues in live animal model to achieve the optimized IntraVital imaging result. And if you have interest in, well, we also provide the basic training service as well for the animal handling for IntraVital imagings. So if you have any question or inquiry, please send the email to us from information@ivimtech.com. And then also to find out the more information, please visit our website, www.ivimtech.com as well. And then if you need, then we can provide this full product and service catalog for you with more comprehensive information inside. So thank you very much for your attention. And then if you have any question, please use the Q&A box, and then I will answer any question you have.